Alright, so in this video we'll be looking at carbohydrates. So what are carbohydrates? Well, we need to take note that carbohydrates are also called hydrates of carbon. So these are simply compounds with this general formula. Okay. These are compounds with this general formula. Alright, so X and Y simply represent variable numbers, okay, or coefficients in other words. We also need to take note that the proportion of hydrogen and oxygen in carbohydrates is in the ratio of 2 to 1, same as in water, okay, because for every water molecule there will be 2 hydrogen atoms and 1 oxygen atom. The same thing applies in carbohydrates. Okay, so carbohydrates can exist in two forms. Okay, they can either be aldehyde, okay, also known as aldoses, okay, or they can exist as ketone, also known as ketoses. Okay, all right. But despite of the form in which they exist, they all contain several hydroxyl groups, the OH groups. Okay. So what are aldoses and what are ketoses? What differentiates aldehydes from ketones? Well, aldoses have a terminal carbonyl group. Okay, yeah, and these are easily oxidized than ketoses. Okay, so these they have a terminal carboxyl group. Okay, these they have the terminal carbonyl group, right? Yes, and they are easily oxidized. So these are easily oxidized, right? Yes, these are not easily oxidized, right? Yes. So since these have got a terminal carbonyl group, it means that these do not have a terminal carbonyl group. And we'll discuss more about what we mean by that as we proceed in this lesson. Okay. So since aldoses are easily oxidized, they act as reducing agents. Okay. And the opposite is true for ketones. Ketones act as oxidizing agents. Okay, yeah. Why? Because they are easily reduced. All right, but we also need to take note that carbohydrates are divided into three main classes. Okay, so we can say classes of carbohydrates. Well, carbohydrates are divided into three classes. Okay, the first class being mono. So I've got mono, then we've got di, okay, and we have got poly. polysaccharide. Okay, we have got the polysaccharides. Alright, so we've got the monosaccharides, we've got disaccharides, and we also have polysaccharides. And their names, okay, all of them, their names end with a suffix. Their names end with a suffix OSE. Okay, yeah. So without wasting much of our time, let's simply jump into monosaccharides. So we'll start with monosaccharides now. Okay, monosaccharides. So want to know what monosaccharides really are. What are monosaccharides? Right? Yes, guys. So what are monosaccharides? Well, there are a few things we need to know about monosaccharides. Okay. First of all, we need to take note that monosaccharides are actually the simplest. So these are the simplest form of carbohydrates. Okay. And they are made up of single sugar units. So they are made up of, so these have got single sugar units. Okay. Single sugar units. Okay. Which are also known as monomers. These are used as direct source of energy. Direct source of energy. Okay. By cells, of course. Right? Yes. So we also need to take note that monosaccharides are classified according to the number of carbon atoms they have. Okay, so the number of carbon atoms they have determines the class in which a certain monosaccharide will fall in. For example, those that have 3 carbons are called trioses. Okay, those with 4 carbons are called tetroses, 5 carbons pentoses, 6 carbons hexoses, 7 carbons heptoses, so on and so forth. Okay, but despite of all those, 3 are common. Okay, 3 are common. What are common? We have got trioses. Okay, we have got pentoses. These are common. And we have got hexoses. So these are common, guys. Trioses, pentoses, and hexoses. Right? Yes. So these can exist either as open chain form or in ring form. So they can exist as open form they can exist as open okay, they can exist as open chain form or in a ring form. Okay, right? So they can exist either as open chain form or in ring form. All right, we'll look at this as we proceed. Okay, yeah. So what else do we need to know? Well, we also need to know that the most common isomer is the D isomer. Okay, yeah, which rotates polarized light to the right. The L isomer is rare and rotates polarized light to the left. We'll talk more about this. Okay, we'll also discuss why the L isomer cannot be digested in our body. Okay, yeah. Okay, well, we are in monosaccharides. Yeah, yeah. We've discussed the types of or the classes of monosaccharides. Yes, the most common forms of monosaccharides are these three. Okay, so among these three, let's start with treoses now. Okay, so let's look at treoses. Okay, so we are now on treoses. Right, so well, we need to take note that there are actually two biologically important treose sugars. Okay, yeah, I've got two important biological treose sugars. The first one being glyceroldehyde. Yeah, it depends with your pronunciation. Okay, glycero. Okay, glyceroldehyde. Okay, so I've got glyceroldehyde, but glyceroldehyde can exist in two forms. Okay, in other words, glyceroldehyde has got two optical isomers. So this has two optical. This one has got two optical isomers. Okay, yeah, which are the L and D glyceroldehyde. Okay, so we've got the L glyceroldehyde and the D glyceroldehyde. All right, then yeah, we said we've got two biologically important choice sugars, right? The first one being glyceroldehyde. Well, what about for the second one? The second one is simply dihydroxyaketone. So the second one is D dihydroxyaketone. Okay, dihydroxyaketone. So by seeing we can tell that glyceroldehyde is simply an aldehyde. Okay, yeah, just by the name itself. Dihydroxyaketone ketone is simply a ketose sugar, right? Yeah. Amazing. Okay, so during glycolysis in cellular respiration, fructose 1 2 diphosphate is broken down into glyceroldehyde 3 phosphate and dihydroxyketone phosphate, producing either lactic acid or pyruvic acid and ATM. Right, so actually, let's take note of that. Yeah, we need to take note of that. What are we taking note of? During glycolysis. So the idea is that in cellular respiration, there is a process called glycolysis, right? Yeah. So in this process, there is this fructose. Let me just write it. It's fructose 1,6 diphosphate. Okay. So this thing is broken down during glycolysis, right? So this is broken down. Okay. So that is broken down to produce what? To produce glyceroldehyde 3 phosphate and also dihydroxyaketone. So this is what happens during glycolysis in cellular respiration, okay, whereby this will be broken down, right, and it will produce glyceroldehyde 3 phosphate and also dihydroxyaketone. All right, yes. So this dihydroxyaketone is called dihydroxyaketone phosphate, which is produced during glycolysis. Okay. Yes. All right. So in this process, okay, lactic acid or pyruvic acid can be produced together with the ATP, adenosine triphosphate, right? Yeah. Okay. Well, let's look at the structure of these two trioses, because we said that we have got this one, which is right here. Okay, we've got this one and this one. Okay, and we said to say this is an aldehyde and this is a ketone. So what's the difference between the two? Well, this is the structure of the two. So we have this. Okay, then okay, all right. So this is This is simply glyceroldehyde. Why do we say so? It's because its carbonyl group is at the end, at the terminal. All right? Yes. Okay. Yeah. Then let's draw the other one, which in this case is the hydroxyacetone. How does it look like? Well, for the hydroxyacetone, its carbonyl group is not at the terminal end. Okay, but rather it is here. Okay, yes. Remember, every carbon is supposed to be bonded to um, 4 other atoms or substances. In other words, one carbon should have got a maximum of 4 bonds, right? For this one, 1 2 3 4. Okay, yeah. What about for this one? 1 2 3 4. Amazing. Okay, so we have our OH here and another one there. On this side we have this. This one is called dihydroxyacetone. What differentiates the two? Well, it's simply the location of the carbonyl group. Okay, as you can see, the carbonyl group in the hydroxyacetone is not at the terminal end, right? Yeah, but for this one, it's at the terminal end. And this one is oxidized. Okay, it's easily oxidized compared to this one. All right. Well, let's proceed to pentose sugars now. Yeah, we've talked about chaoses. Now we move to pentose sugars, then hexoses. That's when we'll be done with monosaccharides. Okay, so we now move to pentoses. All right, so pentoses, just like the name suggests, these are 5 carbon sugars. Okay, yeah, with the formula C5H10O5. Okay, examples of this. Okay, we've got ribulose, ribose, and deoxyribose. Okay, amazing. Well, we need to take note that in this lesson we are going to focus much on ribose and deoxyribose, not on ribulose. But what we need to know about ribulose for now is that it is an important carbon dioxide acceptor during photosynthesis. Okay, so this one accepts carbon dioxide, right, during photosynthesis in plants. You need to take note of that. Okay, what about ribose and deoxyribose? Well, for ribose and deoxyribose, these are simply components of nucleic acids. Okay, yeah, to be specific, ribose is a component of RNA, which is ribonucleic acid, right? Then deoxyribose is a component of DNA. Okay, yeah, which is deoxyribonucleic acid. Okay, amazing. So now we go deep in ribose and deoxyribose. Okay, yeah, you can even call it ribose. Yeah, it's still fine. Ribose and deoxyribose. Okay, well, just like we have already said, yeah, these are 5 carbon sugars. Yeah, they are part of the structure of nucleic acids, which are RNA and DNA. Okay, yeah. So we also need to take note that when carbon number 2 has one OH group and one H atom, the result is ribose. And when it has got two hydrogen atoms, the result is deoxyribose. Okay, that is actually the difference between ribose and deoxyribose. Well, let's simply draw the diagram of the two in order for us to differentiate them easily. Okay, yeah, this is what I mean by that. Okay, so both of them, they have got 5 carbons. Okay, and the other one, okay, okay. Then for this one, if you want, you can write it like this. Yeah, that is, if you want, you can write it in that form, in that manner. Still fine. Okay, because our focus is simply on carbon number 2. Okay, yeah, our focus is on carbon number 2. Well, this is the structure. Okay, so this is what I was talking about by saying that this is our carbon 2, right? Yes. So when carbon 2, okay, when carbon 2 has got one hydrogen and one OH group, the result is ribose sugar. Okay, when carbon 2 has got one H and one OH group, the result is ribose. And when carbon 2 has got one hydrogen atom and another hydrogen atom, this is what I mean. Okay, so when you have got hydrogen this side and hydrogen this side, and this thing has got 5 carbons and it's a carbohydrate, then it is a deoxyribose. Okay, more like it's lacking oxygen. Okay, yeah, because here on carbon 2 there is oxygen, but here it doesn't have oxygen. That's the reason why it's called the deoxyribose sugar, right? Yes. Well, this is ribose and this is deoxyribose. Okay, but this is the D form, yeah, not the L form, and this is also the D form, right? Because this is to the right. Have you seen the OH groups are to the right? That's the reason why it's called the D ribose. When these come this side and like when these and this swap, this will be called the L ribose. Okay, yeah, same thing applies to this one. But we also need to take note that carbon 4 combines with carbon 1 of the open chain of both ribose and deoxyribose, okay, resulting in the formation of a 5-membered furanose ring. Okay, so what simply happens is that carbon 4 will simply combine with carbon 1, okay, to do what? Well, to form a 5-membered furanose ring. Okay, so you need to take note of the name of the ring. It is a furanose. Okay, furanose. Furanose ring, right? Yes guys, yes guys. Okay, so what simply happens now? What simply happens is that the oxygen of carbon 4 will link with carbon 1 to form a closed ring. Okay, so the oxygen, this oxygen will link with carbon 1 to form a closed ring. Okay, yeah, and this can simply be um, expressed diagrammatically. Yeah, diagrammatically. Yeah, it will be diagrammatically shown actually. Okay, yes guys. Okay, okay, is this still clear? Well, yeah, we hope so, we hope so. All right, so let's simply draw the structure of both ribose and deoxyribose. Okay, we draw them in ring forms. Okay, let's look at their ring forms. Okay, all right, so this one can exist in 2 forms. Okay, yeah, so since we have got ribose, this part will be for ribose and this part will be for deoxyribose, guys. Okay, so the structure will be like this. Okay, the structure will be like this. So it will be a 5-membered furanous ring, remember. So it will be like this. Okay. All right. I hope you are able to see, guys. Okay. Are you able to see? Are you able to see, guys? Well, if you are able to see, then we are good to go. Are you able to see? Are you able to see, guys? Right. So we'll start with the B form actually. Okay. So when on this carbon, which is the carbon 1, whenever you see, so here you have got carbon, right? I've not indicated it, but you just need to know that here there is carbon. So here there is carbon 1, guys. Listen, guys. This is carbon 1. Are you getting me, guys? This is carbon 1, guys. Okay. So now on carbon 1, when the OH group on carbon 1 is on top, is like this. When this is on top, just know that this is the B form. Okay? Just know that this is the B form, right? Yes. Well, I'm sure you are able to see this. Okay? So this side will be the outside part, right? So this is the first carbon, right? Which is this one. Okay? What about for this one? So the OH will be down there, right? What will be inside is the H. What about for this part? Well, the same thing will apply. So here we have OH. What will be inside? H. Okay. On carbon 4, we've said to say this oxygen would have reacted, okay, would have combined with this carbon, okay. So here will not have oxygen, okay, but rather we only have H down there, okay. Yes, but what will be put on top, okay, what will be put on top is simply this part, okay, yeah, which can be written as CH2, and this side we have got OH. This is the same as this. Alright, guys, I hope that is clear, guys. I hope that is clear. Okay, so this is called this one is called D-ribose. What is this one called? Well, this one, since the OH group is on top, it is a beta, okay, a beta form, right? Yes. Then it is coming from the D-ribose, right? So we have D, right? D what? It's coming from ribose, right? D, so it will be D ribo. Okay. What is the type of ring that is being formed? Is the furanous ring. So it will be B D ribo furanous. Okay. I hope you guys are clear on that one. I hope you are clear on that one. Okay. Yeah. I hope you are clear on that one, guys. Okay. Then this is the B form. What about for the alpha form? Well, for the alpha form, what we simply swap is this part. The OH group will be down. Okay, the OH group will be what? Will be down. So it will be like this. Okay, so it will be like this, guys. just one and the same thing. Yeah, almost one and the same thing. So this is almost one and the same thing. So on carbon 1, the OH group will be down. Yeah, so on carbon 1, the OH group will be down. Everything else remains the same, guys. Everything else remains the same. Okay, so everything else remains the same, guys. All right, so this is how you draw the ring form. Okay, but let's name this. Okay, if this is beta d ribofuranose, what will be this called? Well, this one will be called the alpha d ribofuranose. What will simply change is this part, because the OH now on carbon 1 is down there. Okay, well, what about for this one? Okay, okay, what about for this one? So for this part now, let's draw its ring form. Okay, let's draw its ring form. All right, so this is simply one and the same thing as well. Okay, the procedure is just the same. Why do I say so? Well, this is the structure. So when the OH is above the group on carbon 1, then it's a B form. Okay, then you go and look at and ask yourself, carbon 2, what do you have this side? So on carbon 2, I've got H there, OH, right? What is inside on carbon 2 is H. Carbon 2, what is in carbon 3, what is inside is H. On this one, this would have reacted, so remain with H. What do we put on top is this part. Okay, are we together, guys? Are we together? Hope this is clear. The only difference, so this will be called what? First, this is a B form, right? Yes, and it is a deoxyribose. Okay, it is a deoxyribose. This is a deoxyribose because these are at this side. When we swap, then it becomes an L, which is not common, right? Okay, so this is BD what? Deoxy, yes. So deoxy ribofuranose. Okay, so this thing is simple, guys. Okay, the only difference between this and this is on carbon number 2, as you can see, because down here we've got the OH group, but here we've got the H. Okay, what other form do we have? We also have got the alpha form, right? Yes, so let's also draw the alpha form. So for the alpha form, it will be like this. So the OH will be down. Okay, everything else remains the same. This will be called the alpha D deoxyribofuranose. All right, guys. So this is it about choices. This is it about choices. Let's now move to this is it about pentoses, right? I was looking at choices here. So this is it about pentoses. We now move to hexose sugars.